खाली सुतली बम घर पे हूँ।
आराम से खोल। क्या जा रही हो, क्या करना है?
आणि मग मी शेअर स्क्रीन अजून तर मला इंट्रोड्यूस करतील ना, मग मला
लेकिन मैडम की स्क्रीन आ रही, मेरी स्क्रीन नहीं आ रही। मेरे को स्क्रीन पे लाओ। स्टार्ट करो। One and all, let us proceed for afternoon session of the today's e-seminar. Honorable Dr. Shamama Parveen, President, Raibar Educational, Cultural and Welfare Society, Principal Dr. Sheikh Kabir Ahmed, and dear participants. Now, next session is of Dr. Meenakshi Mahajan Madam. Dr. Sutar Sangeeta Madam is going to introduce Dr. Mahajan Madam. Sutar Madam, please.
Thank you, Vidya Madam. Good afternoon, everyone. Now I would like to invite Dr. Meenakshi Mahajan Madam. Welcome, Madam. Dr. Meenakshi Madam is working as an Associate Professor, Department of Botany, Fergusson College, Pune. Her area of research is cryptogamic botany, taxonomy, aerobiology, and antioxidants from different plants. 3 students have completed PhD under her able guidance, and 2 students are pursuing for PhD. Dr. Meenakshi Madam has completed 5 research projects funded by different funding agencies. Madam has published 63 research papers in national and international journals. Madam has written 5 books. Madam, she worked as chairman, coordinator, and subject expert on different academic and professional bodies. Madam has visited 11 countries such as Europe, Sri Lanka, China, Malaysia, and Nepal in connection with research work. Now I request Dr. Meenakshi Madam to deliver invited talk.
And share screen.
Dr. Meenakshi Madam.
Can you hear me?
Hello? Hello? Am I audible? Hello, Dr. Sangeeta?
Yes.
Yeah.
Yes. Yes.
Yeah. Thank you, sir. Thank you so much. So, good afternoon, everyone. First of all, I'm very much thankful to Dr. Sangeeta for my nice introduction. And also thankful to the organizers and organizing committee for giving me this opportunity. So thank you so much. So a topic of my talk is role of biotechnology in conservation of plant species. Can you hear me? Yes, all the participants and students?
Yes, madam.
Yeah. Is my screen visible?
Yes, ma'am.
Yeah,
Yes.
Thank you. Thank you so much. Thank you. Role of biotechnology in conservation of plant species. I would like to quote one quotation. I cannot prepare my bun simply sitting in the sun, but the plants have made great fun with the help of chlorophyll gun. So this quotation is by JBS Haldane. All of us know the position of chlorophyll among the living world. So due to chlorophyll, plants occupy strategic position among the living world. All the plants are economically important one, and at ecosystem level, the biodiversity provides all the favorable conditions one. So it's a need of time. It's a need of time to work at interdisciplinary level. So biotechnology and biodiversity linked with each other. What is biotechnology? The biotechnology term coined by Carl Erkel in 1970. Biotechnology is the application of scientific and engineering principles to the processing of matter by biological agents to provide goods and service, or it is the controlled use of biological agents such as microorganisms or cellular components for beneficial use. What is biodiversity? What is biodiversity? Biodiversity is variation of life forms within a given ecosystem. Biome for the entire earth. There are three values of biodiversity: gene, species, and ecosystem. All the three values provide society with a wide array of goods and service. So nature's variation or variety, it is at three levels: genetic variability within a species, variety of species within a community, and organisms of a species in an area. Let us come to the importance of biodiversity. The importance of biodiversity is listed over here: generations of soil and maintenance of soil quality, maintenance of air quality, maintenance of water quality, pest control, detoxification, decomposition of waste, pollination and crop production, climate stabilization, prevention and mitigation of natural disasters, provision of food security, income generation, spiritual and cultural values. Generation of soils and maintenance of soil quality, all of us quite aware with biogeochemical cycle. So all the microorganisms like bacteria, algae, fungi, mites, millipedes, and worms, all they have their great role in biogeochemical cycle because they add crucial nutrients to plants. And the important role of them is to replenish the soil with the new supply of all the nutrients one. Maintenance of air quality, the plants, they have their key role in recycling of oxygen. So all of us quite know the process, physiological metabolism, like the photosynthesis is there, then respiration is there, intake of oxygen, outtake of CO2. So super balance is maintained in the nature. So recycling of oxygen and filtering harmful particles from industrial waste, the plants, they are playing an important role. Maintenance of water quality. Trees and forest soil, they purify water and flows through forest ecosystem. Forests prevent the harmful situations of rivers and reservoirs that may arise from erosion and landslides. Pest control, particularly what? Biocontrol. So all of us know what are the hazardous effects of pesticides. So pesticides, which are chemical in nature, they leave very hazardous symptoms to the human beings. They causes very serious diseases to the human beings. So it's a need of time, or we should give more emphasis on the use of biocontrol. So from that point of view, it is very essential to study biodiversity. The next importance of biodiversity is in the detoxification and decomposition of waste. So organic waste processed every year by Earth's decomposing organisms. So decomposed industrial waste, detergents, oil, papers, acids, or at the end of this process, simple inorganic nutrients, they are formed. The next importance, pollination and crop production one. So the organisms, all bacteria, bees or butterflies or birds, they facilitates pollination on which the humanity or food crop is dependent one. Besides this, the dispersal of seed mechanism also has very important role. The next importance is climate stabilization. The plant tissues and other organic materials within land and ocean ecosystem act as repositories of carbon. They utilize CO2 in photosynthesis, contributing to climate stabilization to contrast the surface or to control the surface temperature. Forests play an important role in controlling temperature. So cold climates, forests acts as insulators and they maintain what? Cold climate, helping to mitigate the impacts of freezing temperature. Next importance is in prevention and mitigation of natural disasters. So forest and grassland protect landscape against erosion, nutrient loss and landslides through the binding action of roots. Forest and grassland ecosystem help to absorb excess water and thus reduce the damage caused by floods. Besides this, the coastal ecosystem, they also prevent erosion of coastal lines. Next importance of biodiversity is in provision of food security. So plants and human welfare, that is that part we study in economic botany. So all of us know the different aspects of plants, plants that feed us, plants that cure us, the plants that feed us, which we use as food, plants that cure us, that refers to medicinal plants, then plants which are used as spices and condiments, plants that used as cup and cheers, they are all the beverages, then plants which are used in perfume and cosmetic industries, then the lower plants in service of man. So in industries or in agriculture, the algae, fungi, bryophytes, they are used as fertilizer, or they are used as biocontrol, or they are used for maintaining fertility of soil. Bryophytes, they are used in reclamation of soil. So it's a need of time to give more focus on the study of lower plants. Next importance is in income generation. So biodiversity has great importance as a direct source of income and economic development. Bioprospecting involves characterization of bioresources, the different methods or exploration of wild plants for commercially valuable genetic and biochemical resources. So the biodiversity of such microbes in pharmaceutical industries, in fermentation industries, in antibiotic industries, enzyme technology, toxins, vaccines, hormones, alkaloids is very promising and challenging task. So biodiversity give great service through all ecotourism one. And the last importance, that is what spiritual and cultural value. So all of us know about ethnobotany. In ethnobotany, all the traditional uses of plants has been given or described one. So it is very important to know about the ethnobotanical information of all the existing plants on the earth. Why? Because to go for certain formulations in pharmaceutical industries. So from that point of view, we can take some base of ethnobotany and we can go or we can do certain revolution in the field of pharmaceutical industries or in the field of pharmacology or in the field of medicinal plants. So from that point of view, it is very essential or important to know about our cultural values. There is an umbilical bond between humanity and biodiversity reflected in the art, religious and tradition and tradition of diverse human cultures. Why to conserve biodiversity? To protect the genetic resources, conservation of microorganisms helps in reclamation of wasteland and revival of biological potential of land. Ecological stability and productivity, to fulfill the food requirement, to meet most of the basic needs, to minimize the rate of pollution, rehabilitation and protection of endangered species, and defragmentation of forest areas. So ecosystem stability and productivity, the life of organism linked to ecological services. So ecological services like soil erosion, like photosynthesis, nutrient cycling, and food production. So one species or a few species, if eliminated, does not create any problem. But when it is not creating any problem, provided that some alternatives are available in the nature itself. So alternatives are there, and the work always, the environment always work with all such type of alternatives. We are quite familiar with the food chain, food web, everything. Thus, biodiversity imparts ecological stability. If there are no such alternatives, there won't be any stability in the ecosystem. So for the proper functioning of, or for homeostasis of ecosystem, alternative should be there. And productivity, which work at two levels, that is multiplication of species, which refers to biotechnology, and the agriculture one, the agriculture scientists, they are working on crop variety or wild variety, and they matches production technology with the production one, and matches production technology with the production one. Why it is essential to match both the technology with each other? When both the technology get matched with each other, then only we can get increase in yield. So our basic aim, our basic motto is to increase the yield, to increase the production with protection, less expenses on protection. So that is what the concept of agriculturist. Then rate of to minimize the rate of pollution. All of us aware with what the role of indigenous plants in the environment. Nowadays, municipal corporations making compulsion to go for plantation of indigenous trees, because it's a gift of nature to the plants. It's a gift of God to the plants to encounter the difficulties in the pollution. So they possess such type of machinery which can overcome the harmful effects on the harmful effects of the pollutants. So whatever harmful air pollutants are there, they are settled on the leaves of plants, and with the great physiological metabolism, the plants can encounter the difficulties, solve all these problems in their metabolic cycle, and they survive. So from that point of view, the municipal corporations, they are making compulsion to go for what we can say, the gardens and the municipal parks or the gardens in the heart of the city, so that the plants can minimize or reduce the rate of pollution. One, rehabilitation, protection of endangered species. This is very important as far as Red Data Book concerned, because many of the species over the globe, they are becoming endangered, they are becoming vulnerable, extinct, critically endangered. So it's a need of time that all the species should be available not only for this generation, but for the future generation as well. So from that point of view, the biotechnology is playing an important role in conservation of such type of species, defragmentation of forest areas. Yes. Therefore, biodiversity must be conserved for their sustainable utilization through an integrated and scientific oriented action program. This should involve aspects like protection, preservation, maintenance, exploration, conservation, and sustainable utilization. So it is very necessary to protect the biodiversity. It is very necessary to preserve the biodiversity for the further future generations. It is necessary to maintain the biodiversity, and it is necessary to go for exploration of the biodiversity over the globe. Because sky is the limit. Sky is the limit where many areas are unexplored over the globe. So still scope is there to go for doing some revolution or research in the field of biodiversity. So that is what exploration, conservation, and sustainable utilization. So introduction and application of methods and processes for the utilization of biodiversity to prevent its long-term decline. That is very important. Long-term decline depends on how well the environment impact assessment carried out, how conservation strategies operate. So for the future generation, the biodiversity, the use of biodiversity, it should be from sustainable point of view. So sustainable utilization is very important one. Yes. Next is what? The illegal practices for personal benefits. Why to conserve biodiversity? It is to be conserved for to illegal practices for personal benefits. So biodiversity contributes in many aspects of human well-being by providing raw material and contributing to health. The Millennium Ecosystem assessment shows that human action often leads to irreversible losses. So man is only becoming what? Interfering element. So due to urbanization, industrialization, the man is disturbing biodiversity. So the Millennium Ecosystem assessment shows that human action often leads to irreversible losses in terms of diversity of life on Earth. Yes, lack of knowledge, information, and awareness in public sectors, then exploration of areas for documentation. This is very important. What? Documentation one. So documentation, why it is important to maintain the record. So it should not only be linked to the catalog, but it should be linked with habitat destruction as well. Then we should go for preparation of database. We should go for preparation of database. So database, ideally, the database should be linked to network of local, national, international levels that make information accessible. To maintain all the record of biodiversity, long-term monitoring is required. So long-term monitoring required to link the data of past, present, and future. And why to conserve biodiversity? To maintain the socio-economic development. To maintain the socio-economic development one. Biotechnology, which is developed in two phases, classical biotechnology and modern biotechnology. So classical biotechnology, that is kitchen biotechnology, preparation of curd, ghee, vinegar, alcoholic beverages, jalebi, idli, dosa, uttappa. It is based on capabilities of what? Microorganisms one. And the modern biotechnology, which particularly deals with introduction of new techniques which are meant for conservation purpose. So this particular aspect, the modern biotechnology, it works at interdisciplinary level. So biotechnology is one of the branch which is linked to the biodiversity. So if we would like to protect the species, after knowing the red data book status of the plant species, if we would like to conserve them for future generation, we should concentrate on the biotechnological aspects. So biotechnological aspects basically, or rather we can say that the conservation aspects of any plant species can be carried out in two ways, that is in situ conservation and ex situ conservation. In situ conservation. In situ conservation, this type of conservation applies only to wild flora and fauna and to the not to the domesticated animals and plants, because conservation is achieved through protection of population in nature. So in situ conservation is that conservation which is to be done in natural condition. So it is applies to wild flora and fauna. So it can be achieved through protection of population in nature and national parks, sanctuaries, nature reserves, natural monuments, cultural landscape, biosphere reserves, fish genetic resources, fresh water, marine ecosystems, mangroves, corals, and islands. All they are meant for in situ conservation. So we can bring or we can brought in situ conservation by management of protected areas. Management of protected areas. The protected areas, they are in the form of all like national parks or monuments or the landscapes or the biospheres, biomes or the various types of ecosystem. So at priority, they should be maintained, they should be managed very well. Then second thing is habitat restoration and recreation. Habitat restoration and recreation. And the third one, to promote sustainable plant use and land management. To promote sustainable plant use and land management. So this everything about in situ conservation. So in my talk, I have included some of the aspects of ex situ conservations only. Yes. So all these are what? The technological aspects used for ex situ conservation. So the traditional method is there, that is conventional methods by seeds. We can go for collection of seeds, we can maintain the seeds. Different methods are there, the different methods of drying are there, different methods of storage are there. Then the biotechnological aspects of ex situ conservation involve seed gene bank, tissue culture gene bank, pollen and spore bank, cryopreservation, botanical garden, genetic engineering, and nurseries. So I have taken somewhat in detail about cryopreservation and tissue culture gene bank. We have next lecture on tissue culture. So we'll study everything about tissue culture in that lecture. So let us come to what? The seed gene bank. Why do we have seed banks? Why do we have seed banks? The storage of material in the form of seeds is what? One of the most widespread and valuable approaches to conservation. So seed banks, they are most widespread and valuable. We can say that the method of seed bank, the method of conservation by seed bank is advantageous over other methods of ex situ conservation. Now the question comes, why they are advantageous one? they are advantageous, or it is advantageous as its ease of storage, economy of space, relatively low labor demands, capacity to maintain large samples at an economically viable cost. Economically viable cost. So these are what all the advantages of seed bank. Why store seed banks, seeds and not whole plants? So seeds, they are small in size, they are easy to handle, they require low maintenance, they remain viable for long period. This is very important because the viability of the seeds play an important role in germination part. So they can remain viable for longer periods, and as often they require less space. Therefore, we are to store the seeds instead of whole plants. So seed gene bank, in the figure, in the image it has been shown. After collection, the seeds, they are transferred to a gene bank for storage. The samples, they are dried and they are stored at sub-zero temperature. They are stored at what? Sub-zero temperature. So all these are what? The advantages: seed storage conservation, advantages, efficient and reproducible, feasible for medium and long-term secure storage, wide diversity of each target taxon conserve, easy access for characterization, valuation and utilization, little maintenance once the material is conserved. So all these are advantages of what? Storage of seeds. Earlier we have discussed advantages of seeds, and these are advantages of seed storage. As each coin has two facets, likewise for seed gene bank also, advantages are there and disadvantages as well. So let us come to the disadvantages. Problem storing seeds of recalcitrant species. Recalcitrant species, then freezers, evolutionary development, then genetic diversity may be lost with each regeneration cycle. Problem storing seeds with the recalcitrant species. So depending upon moisture requirement during storage, tree seeds are classified into two groups, namely orthodox seeds and recalcitrant seeds. Or depending upon what? The moisture content. When the moisture content less in the seeds or it is low in the seed, they are often referred as orthodox seeds. And recalcitrant seeds are provided with high moisture content. So if you would like to store them if you would like to store them, the storage life, it depends upon their moisture content. So it is taken into before storage of seeds in seed gene bank. It should be taken into consideration what type of the seeds are, because all the methodology, they are not applicable to all the plant species, because angiosperm is one of the group in which we get lot of variation, lot of diversity. So with respect to consideration of this diversity, we should consider about the seeds of plants with reference to moisture content. Yes. Next aspect of ex situ conservation is what? Before that, one more thing I forgot, sorry. The seed storage method, the storage should be at ambient temperature and humidity. The dry storage with regulated moisture, dry storage with controlled moisture and dry and temperature, then cryopreservation, in running water or under potential vacuum, and coating of seeds with paraffin or latex to prevent moisture exchange. All these methods, they are used for seed storage one. The next method of field gene bank, ex situ conservation, is field gene bank, collection of seed or living material from one location and its transfer and planting at second site. Large number of accession of available. So a few species are usually conserved. So field gene bank, it is advantageous over the storage one because it is suitable for storing material of recalcitrant species. So according to the type of seeds, we should go for selection of method of ex situ conservation. So easy access and it is advantageous because it gives easy access. It made available easy access for characterization, evaluation, and utilization. So again, some disadvantages are there for field gene bank. That is, material is susceptible because material is susceptible to the attack of microorganisms, causes diseases and vandalism. Vandalism is what? It's a destruction. So all the microorganisms, they causes serious diseases to the seeds. Many seed-borne pathogens are there, which cause seed destruction. So it involves large areas of land, but even then, genetic diversity is likely to be restricted one. And it require what? High maintenance cost. So maintenance cost should be taken into consideration when we go for selection of ex situ conservation method. Botanical garden arboretum. Botanical garden arboretum. Seeds transfer to second location as living plant collection of species in a garden or arboretum. So to store the seeds in the botanical garden, that increases what? The shelf life as well. So it is advantageous one, freedom, because it gives freedom to focus on wild plants and economic plants. Because the botanical garden or arboretum, they mainly they are the main places which are very actively involved in conservation of wild flora. It's a need of time to go for conservation of wild flora, so we can superbly conserve wild flora in the botanical gardens. Disadvantages are also there because space is one of the limiting factor for that. Then genetic diversity of the species may not be conserved sometimes. Involves large areas of land, high maintenance cost in glass house. High maintenance cost is there to conserve them in glass house. Yes, excuse me.
Yes, madam.
Yeah, yes, just a minute, madam. Thoda net problem is there. Just a minute.
Okay.
Okay, no problem, ma'am. No problem.
Can you hear me? Hello?
Yes, ma'am.
Yeah.
Ma'am,
Yeah.
yes.
The next method of ex situ conservation is DNA or pollen storage. So collection of DNA or pollen and the storage in appropriate refrigerated conditions. So this method also has got some advantages. the advantage that is relatively easy, low cost of conservation, and it has got one more advantage that that is what we can handle number of samples at a time. So as the huge sample of DNA available, we can make them in a small fraction or a small segment. So the samples are more available. Disadvantages of this one: DNA regeneration of entire plant from DNA cannot be envisaged at present, and we encountered problem with subsequent gene isolation and cloning transfer. Pollen, which are the male reproductive organs, they need to develop individual regeneration. So protocols required to produce haploid plants, and further research needs to produce diploid plants. So only parental material conserved, but mixture from many individuals could be envisaged. So this shows what the advantages of pollen. So pollen, the role of pollen in pollination and hybridization. So hybridization means giving new varieties of the plants. So when we go for this, we can raise individuals. So plants that flower at different, all these are what the advantages for. So plant flower at different times, which mature at different times. Then we can go for study for allergens, biochemicals, and physiological studies. And international exchange of germplasm should be there. We can go for gene conservation. We can go for recalcitrant pollen and haploid production, and then diploid one. So hybridization between plants with flowers at different times. That is what the advantage of pollen development. Growth at different places, reducing the dissemination of diseases by pollination vectors, and maintenance of germplasm and enhancement of longevity. Now the next method, which we'll see little bit in detail, that is what? Cryopreservation. So the word cryos come from the Greek for ice, frost or cold. Preserve means to keep safe from harm or loss. Cryopreservation is the process of freezing living material to keep it safe. So as far as in situ conservation concerned, everything depends upon what? The nature. So man has realized there should be some revolution in the field of conservation. So we can link conservation of the plant species by using biotechnological techniques. And one of the popular or common technique is cryopreservation. Cryopreservation is the process of freezing living material to keep it safe. So it is what a living material. The material, it is freezed and then it is kept in the liquid nitrogen. The temperature of liquid nitrogen is minus 190 degrees centigrade to 320 degrees Fahrenheit. So the question may come in our mind, why have this range of temperature only? So this temperature, physical and metabolic cellular processes are effectively, and living tissues, they are in state of suspended emission. They are free from pathogen, and risk of genetic drift is not there. So cryopreservation, in theory, plant tissues can be stored in this way indefinitely when required, and it can recover and grow into generate a whole plant. So there are three principal methods used for. There are three principal methods used for growth suppression in plant tissue culture. These three principles of plant tissue culture are the alterations of physiological conditions in conditions of culture, that is temperature or gas composition within the vessels. Second, changing the composition of vessel medium, example, using such or some optimal conditions of nutrients. And third one, supplementing the medium with growth retardants like acetic acid or osmoregulatory compounds like mannitol, sorbitol, etc. Storage at reduced temperature has been very effective for tissue culture of species like potato, cassava, pea, chick, focus, etc. Now let us come to the methods of cryopreservation. So, method of cryopreservation includes how many steps? There are 7 steps. The 7 steps, namely choice of plant material, pre-treatment, freezing, storage, thawing, post-treatment. There are 6 steps are there. Choice of plant material. Selection of plant material is very important as far as cryopreservation is concerned. Which material is most suitable for cryopreservation? The material which is which contains amount of cytoplasm, which do not possess vacuoles, that do not presence of large number of vacuoles, and which can tolerate freezing temperature. These are the basic conditions for the selection of material 1. Second step is pre-treatment. Pre-treatment because a lot of variation in the nature. So all the plant species, they are not in need of the same type of treatment. We should first of all we should know about the plant species and what type of treatment should be given. So, due to variation, it should be clear to the scientists how is the status of dehydration of the selected material. Whatever the material is selected, it should be able to stand the cryoprotectors for dehydration of the tissues and for protection of cell membrane. Third step is what? The freezing one. That will study that the storage, the thawing, post-treatment one. So the third step, that is what? Freezing one. Theoretical basis of what? Cryopreservation, which includes freezing. Why to store or why to freeze the material at -196 degree centigrade? So it is very clear to us what the material should, that should not be intracellular accumulation of ice crystals in the plant material one. From that point of view, what we have to maintain the temperature 1. So this storage at -190 degree centigrade are influenced by the ability of the material with respect to morphological and physiological conditions of material, except the plant used. Certain organic, certain structures are there, like apical meristem, ovule, anther, polar, embryos, protoplast. They can stand very well. We can select such type of material. At which stage of growth, the suspension of many plants has successfully been cryopreserved. The cultural cells, they are not ideal system that can be preserved. So organized structures like shoot, apices, young plantlets, embryos preferred for cryopreservation. Can you hear me? Yes? Yes. Hello. Am I audible, sir? Am I audible?
Yes, ma'am. Yes.
Yeah. Yes.
Yes,
Then,
ma'am. You are audible.
yeah.
Yes.
Second step is what? Addition of cryoprotectors. So after selecting the material, then it is ready for freezing. And freezing can be done in what? In cryoprotectors. So sugars, glycols, alcohols, polyvinyl pyrrolidine, polyethylene glycol, abbreviated as PG, polyethylene oxide, PO, dextrose, hydroxystarch, glycerol, 10 to 20%, and some amino acids, and DMSO4, that is diaryl sulfoxide in 5 to 10%, used as cryoprotectors. So during cryopreservation, two strong sources causes cell damage. The first causes formation of large ice crystals inside the cells. Intracellular formation causes destruction of cell organelles and the cell itself. And second cause is an intracellular concentration of solutes that increases to toxic levels due to dehydration before or during storage. So these are two causes which bring damage of the cells. So all these cryoprotectors, they are added. Why? Because they decreases cryo destruction. They decreases cryo destruction. Now it has been advised how to use the cryoprotectors. So it is advised to use a mixture of 2 or 3 cryoprotectants at low concentration rather than a single cryoprotectant at higher concentration. If we go for the use of single cryoprotector in high concentration, we increase the risk of destruction of the cells, destruction of cell organelles. So it has been recommended or it has been advised that whatever use of cryoprotectants, it should be in combination of 2 or more. The commonly used cryoprotector is DMSO4, that is dimethyl sulfoxide. Then sucrose is there, glycerol is there, proline is there. DMSO4, sucrose, glycerol, and proline, they are commonly, or rather we can say, they are good or excellent cryoprotectors. They are good or excellent cryoprotectors. Why? Because it has, they have low, the DMSO4 among sucrose, glycerol, and proline. It is good because it has low molecular weight. It is easily miscible with solvent. It is toxic at low concentration. It is easily permeable into cells and easily washable. Easily washable. That's why it has been suggested that, or it has been advised that DMSO4 should be given at priority. So material which is to be preserved is put in culture medium and then it is treated with cryoprotectant. So such material is transferred to sterile cryovials, which are made up of polypropylene, and gradually 5 to 10% of cryoprotectant is added into the vials and then closed with a screw cap. So this is what the procedure of cryopreservation. Once we have supplemented the material in vials, it is ready for what? Storage. So freezing, it is at 3 levels. Rapid freezing, slow freezing, and stepwise freezing. So it does not, freezing does not cause intracellular freezing. and crystal formation inside the cell. To avoid this problem, regulated rate of cooling or pre-freezing is done. So freezers have also been developed which allow the uniform temperature decrease at a desired rate. This is very important. The freezer should have a uniform decreasing rate of temperature. Then only the plant material can superbly store in liquid nitrogen at minus 196 degrees centigrade. Let us come to the first method of freezing, namely rapid freezing. Rapid freezing, it's a simple and easy to handle method. The plant material in cryovials, it is put into liquid nitrogen, which causes what? Decrease in temperature, which causes decrease in temperature. Freezing is done quickly so that there should be least change or development of intracellular crystals. Ultra cooling required for this. Why? To prevent ice crystals. To achieve this objective, dry ice can be in the form of CO2 can be used instead of nitrogen. So rapid freezing of several plant material has been done. For example, somatic embryos and shoot tips of Brassica napus, strawberry, potato. Second method of freezing, slow freezing. What is the required temperature for this? Temperature, decrease in temperature from or temperature of 0.1 to 10 degrees centigrade per minute. So this indicates the flow in water from inside to outside. Therefore, extracellular ice crystals are formed, but not intracellular crystals. So cell cytoplasm gets concentrated to cause cellular dehydration. Such dehydrated cells survive for a longer duration, and generally slow freezing has been done by using computer controlled freezers. Then stepwise freezing is there. So stepwise freezing includes what? Decreasing temperature, the temperature lowered by minus 20 to 40 degrees centigrade. And this method bring or concerned with the formation of big crystals, which is a good result for this, and it's excellent results that have been obtained for suspension cultures and strawberry by using this method. So under freezing, three methods are there. Storage in liquid nitrogen. So that is very clear to us. Why to store material at minus 196 degrees centigrade? To stop all the metabolic activities, to prevent biochemical injuries, and especially it has been carried out for the carrot cells for 5 years. So we can we can superbly store or culture of carrot cells stored by this for 5 years. The next method is what? Thawing. THAWING, thawing, that is defrost. Cryovials kept in warm water bath at 35 to 45 degrees centigrade. So releasing the vials from frozen state to elevate the temperature between 35 and 45 degrees centigrade. It is done and by overheating. So as soon as the last crystal disappear, the vials are transferred into water bath maintained at 20 to 25 degrees centigrade. And the last step, the second last step is what? Washing and reculturing. Why? To remove the toxic cryoprotectors. This step involve dilution, resuspension, centrifugation, and removal of cell. And the last is what? The regeneration of viable cells in plantlets. The regeneration of viable cells in plant cells. So viable cells, they are cultured or non-specific growth media to regenerate into plantlets one. So this is everything about what? Cryopreservation. So we encountered certain difficulties in cryopreservation. The difficulties in cryopreservations are high specific features of plant cells, such as their size, strong vacuolization and abundance of water, then cell dehydration, cell damage during freezing and subsequent thawing caused by ice crystal form inside the cells, then gradual formation of large crystals of more than 0.1 micron whose facets rupture many cell membrane, and there is marked dehydration and protoplast shrinkage. Protoplast shrinkage, that is plasmolysis 1. So excessive time and degree of plasmol are the reasons of cell destruction. So these are what the achievements through cryopreservation, and we have discussed what the difficulties of cryopreservation. So we can take some message from this talk. What type of message is there? Intensive bioprospecting program should be taken up to identify the exploitable resources based on ethnobotanical information. Then a powerful synergy between intellectual property and rich biodiversity, which has to be created. Then legal system has to be tightened up for protection, transfer of technology, and acclimatization of plant species from their original habitat. Because when we go for doing certain experiments from wild habitat to the laboratory one, acclimatization of those wild species to the laboratory or artificial condition, it is compulsory one. So from that point of view, initiative should be taken. Then the selected species should be studied for ecological, phenological and other aspects to develop agrobiotechnology for their cultivation in other areas, and export of finished and valued added products. Yes, so this table particularly highlights about the status of plant diversity at Fergusson College campus. So Fergusson College campus has taken conservation initiatives, and the project, it has been completed by me from 2004 to 2007, until we are revising all the data. So this is what the data of campus: total number of native species 98, total number of exotic species, and all these things. Then media and newspapers, they have highlighted this because it was a pioneer type of work in Pune. And during this project, I have listed some of the species which are endangered one, and started cultivating those species with the help of tree plantation program. And this is what ultimate message in the form of fresh codes. So thank you very much for this talk. I'm very much thankful to the organizers.
Discussion मुझे सर में मुझे सर में दिसत नहीं. Thank you, madam, for your informative presentation. Now, session is open for discussion. Is there any question? I think no question. Thank you. Thank you, Meenakshi madam, for delivering lecture on. Madam highlighted importance of biodiversity, conservation, seed bank, gene bank, botanical garden, pollen storage, and cryopreservation. Madam, your lecture will be helpful to all of us. Thank you, madam, for your nice presentation. Thank you, one and all. Now,
thank you so much.
I request Sangeeta Ahuja madam to conduct further session. Sangeeta Ahuja madam.
Now, it is an honor and pleasure for me to introduce and welcome the great personality in the field of physiology and Plant Tissue Culture, Professor Narayan Pandure, sir. One of the best mentor and our guide. Words are few to speak and about him. Professor Narayan Pandure, sir, is working as a professor in Department of Botany, Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. His area of research is plant physiology and plant tissue culture. He is having 16 years of teaching and 22 years of research experience. Under his able guidance, 3 students completed MPhil and 9 students completed PhD, and 6 students are pursuing PhD. Sir has published 70 research papers in national and international journals. Professor Pandure, sir, organizes workshops on tissue culture and training programs for students and farmers. Sir is expert in setting tissue culture laboratories. Sir has worked on different academic and professional bodies as chairman, member, and subject experts. Sir has received prestigious Ideal Teacher Awards from giant international organizations. Now, I request Professor Pandure, sir, to deliver his talk. Professor Pandure, sir. Professor Pandure, sir.
Correct.
Pandure, sir. I request Professor Pandure, sir.
Pandure sir, please start your lecture. Yeah.
Unmute yourself, Pandure sir.
Pandure, sir.
त्यांचा माईक सुरू नाही.
Your mic is off, sir. Yes. Yes, sir.
Thank you. Thanks to the organizers. जर मोबाईल आणि लॅपटॉप चालू असेल तर मोबाईल दूर ठेवा. Great reformer. सावित्रीबाई फुले.
सर तुमचे मोबाईल आणि लॅपटॉप दोघं सुरू आहेत.
सर दोन्ही चालू असतील तर मोबाईलचे बंद करा, लॅपटॉपचं राहू दे चालू.
Yeah, there is echo, sir.
इको आवाज येतोय सर.
ते लॅपटॉप वर पण आहे आणि मोबाईल वर पण आहे.
हम्म.
त्यांचं कुठलं काय
हे करा आपल्याला करता येतं का?
ते लॅपटॉपचं हे चालू करा म्हणतात.
माईक का?
हा आणि याचा बंद करायचा.
फोन करू का त्यांना?
हा पंडोरे सर तुमचं
मोबाईलचं बंद करा.
मोबाईलचं बंद करा, लॅपटॉपचं सुरू ठेवा. माझ्या लोक
आता येईल का आवाज?
हा बोला ना सर. आता ओके.
ओके ना?
येस सर, येस सर.
It's better now.
You can proceed, sir. No problem.
प्रेझेंटेशन दिसते ना?
Yes, sir. Yes, sir.
So I am very much thankful to this convener of this conference, Professor Milind Jadhav sir. I congratulate to their companions, Ahuja madam and Sangeeta madam. This is a great occasion to talk on this. Today I am talking on the biotechnology for conservation of biodiversity. Basically, whenever we are talking about the biodiversity, then we know that this is the universe which has been created by a creator, and we always praise him that Mangalam Bhagwan Vishnu, Mangalam Garuda Mangalam Sri Kundalikaksha Mangala Hari. As we know that why we are calling this, because this system is so complicated. Even though there is a diversity, but there is a uniqueness in this system. And then secondly, as we are the learner of the botany, then we can say that we can see the wealth of the plant. This universe is there, it is rich in the this plant system. And then we know that these plants are there, they are very much important. And regarding plant, it has been said in Upanishad that Amulam Aushadam Nasti, Amantram Aksharam Nasti, Ayogya Purusha Nasti, Yojaka Tatra Durlabha. So as we can say that this each and every plant is there, it is important for us, and then we are going to take advantage of these plants. Secondly, we know that so this plant wealth is there, and then apart from if we are watching the commercialization, then we can see that these Kerala peoples are there. They have taken a good advantage of this system, and a real commercialization has been taken place. Secondly, we can also talk about the Baba Ramdev, and we can see how much he is sharing more than 40,000 crore rupees, or that much his investment is there in this Ayurvedic system, and he is earning a lot through this. So looking toward this, we can say that this system, these plants are there, they are valuable. Then today we are talking about, as we are talking about the diversity, we know that it is a variability among the living organism and ecological complexes in which they occur. So this biodiversity is there. It is of three types, as we know. I have not to go through the detail, but it is a genetic diversity is there. Then species diversity there, and ecosystem diversity is there. If we are looking towards the global diversity, then we can see that about the 56% insects are there, 14% plants, and 3% birds and mammals are there, which has been among the 1.8 million species. And when we are looking towards this diversity, then we see here that the number of this which has been identified. Here is the table. You can say that total number of species and then number of species identified. So huge and large number of this diversity there. It is occurring in this universe. Then apart from that, we can say see that this diversity there, it is important for us, and we have to take steps for the conservation of the diversity. Here we are looking at a slide, and in this we can see that the diversity of the plant. See the Rafflesia. Even though the roots are here like, but a big flower is there. We can n number of orchids are there, orchid which are beautiful one. So we are going to see the orchids. Then we can see the flowers here. These are the important flower we can see. Then insectivorous plants, plant diversity is also there. Then we can see another. Apart from that, among the hotspot of the diversity, we can see the hotspot of the diversity, and we know that India is there one of the best hotspot in which you can see the Western Ghat is there, which is rich in the diversity. And if we have seen the endemism, that apart from this plant species, more than apart from the taxonomic group, more than 3,000 plant species are there, which are endemic in this area. A huge number of diversity there. And then here you can see that certain flowers are there. Nilkunj, you can Strobilanthes is there, or some mammals are there, animals are there, which are here in the diversity. And then we can see that the diversity there, it is in danger. Several reasons are there for that, among which urbanization may be there, industrialization may be there, or agriculture, pollution, natural calamities, small population, habitat destruction, exotic species, and over exploitation are the several reasons through which the diversity is there. It has brought into in danger. If we have to conserve the, then why we have to conserve the diversity? Then we have to conserve it. If you can see in the slides that, and in those mass extinctions, we can see the number of diversity that it has been vanished. In the, for example, in Ordovician era, in 4,050 million year ago, about 20% of these families were there destructed. Then in Devonian, Pro-Triassic and End-Triassic, and at the last, we have seen that about 90% of the this diversity was lost. And this is the current situation or scenario in which, due to the human impact through habitat destruction and other means, the destruction is going on. We have to take certain measures. Then we can see that the habitat loss is going to take place. Then invasive species are there. So we can see n number of species which are invasive one. Say for example, we can Gliricidia. We can call it as Undirmari. So such species are there which has been occurring, and they are destructing our day-to-day these plants and animals. For that conservation of the diversity, then apart from that invasive spacing, it has been called that Bagh bachega to Jangal bachega. Here in the slide you can see that poaching is going to take place, and due to that poaching, these animals are there. Tiger may be there, elephants are there, leopards are there. Nowadays you can see that leopards are there. They are started to come in the localities of the human, and due to this this destruction of the diversity is going to take place. So we have to take the measures for the conservation of the diversity. Then another reason is there, pollution is there. Then the accumulation is there. Now these farmers are there. They are utilizing pesticides, DDT, and due to this, this flora and fauna or protozoans are they are coming into endage. Then climate change has been taken place, and due to that climate change also, several losses of the diversity has been taken place. And you can see certain slide that due to this climate change, tsunamis are there, they are going occurring, or hurricanes are there occurring. And here in the slide you can see that the impact of tsunamis before and after. Then we can see that the floods are there, we are occurring and taking place. Forest fires are there, volcanic eruptions are going to take place. So these are the, and then we have to save this diversity, and for that conservation of the diversity we have to take certain measures. And then here we can see that tree of the biotechnology, in which we can see that it has got recently Mahajan madam has very nicely presented her presentation, and several aspects of this biotechnology has been shared by her. Then for that sake we have to do certain measures. and we have to do certain with the plant. But as we are the botanist, we have to go for the plant for the conservation of the diversity. We have to then we are after the changing the plant. Now we are going to see. So why we are going to change the plant? Because we need more for population. The food is needed. Then according to change in environmental condition, we have to change the plant also. Secondly, we have to bring hybrid vigor into those plants, and lastly, to provide food, clothes, and shelter, which are the primary needs of the human being that we have to give. And then for the to improve the plant, various methods are there. It may be introduction, selection, hybridization, mutation, tissue culture, and genetic engineering. In the introduction, we are going to see that we are going to introduce the plant species from external regions, and then we are going to introduce in there. Selection is the proper selection of the plant. The pure or superior quality of the plants are they have been planted, and inferior qualities are they have been rejected. In hybridization, good quality of the plants they have been brought together, and then then in mutation, sudden change, we know that sudden change in genome, then due to mutation also we are going to change the plant type. Then here tissue culture, and lastly genetic engineering. So these aspects are they have been handled for the to change the plant. Today I am going to talk basically on the plant tissue culture. Each one is knowing the plant tissue culture, but as I am the learner of this, I am not the expert, but I am learner of this plant tissue culture, and just I am sitting with these cultures. You can see that here these are the cultures of Taxa pinnatifida. Second, you can see the cultures of this Shiropegia bulbosa. One of our student is working on 3 species of this Shiropegia bulbosa. Then secondly, you can see here the cultures of this Taxa pinnatifida. You can see that root formation is taking place in utroculture. And so I am learning each and every day. When I visit to my laboratory, then I can see I am going to watch them carefully, and new information that it has been added in this field. So as it is a plant tissue culture, but it is a culture of plant tissue and cells, and collectively it has been called as plant tissue culture. And with this technique, we can propagate the plants on a large scale in this cultural condition. This tissue culture technique, as we know that the totipotency is the principle on which it has been based. The toti means total and potency means ability. So it is an ability of plant cell to regenerate into entire plant. So with the help of this plant tissue culture, we can grow the plant. So we are going to take as a single cell, or we are going to take an organ, or we can take and then we are growing it into entire plant. So for that plant tissue culture, then we are not to go through the detail, but we know that the Schilden and Schwann in 1839 they have proposed, and they told that if we have done this, or if we have taken plant cell and we have given a proper environment to that, then we can grow these plants into so with that. So by taking advantage of the technique, this plant tissue culture was initiated, and nowadays and n number of plants where you have the tissue culture with the advantage of this technique. Now so with this we can say that this tissue culture technique has brought a revolution, and it has revolutionized. And then here the person Gottlieb Haberland, he had done the pioneer work on the plant tissue culture, and he work on plant tissue culture, and given first of all he had done this initiated this tissue culture. Then here Apart from that, here are the Miller and Cook. They are here. You can see that these are the persons or fellows who had brought first time the excavation of this carnitine or growth hormones were there. They have been invented from the Clupea heringis, or it is a herring sperm DNA, and with the help of that growth hormones, then we can do the plant tissue culture properly. It was Stewart in 1958 that he had done the carrot tissue culture by taking a cambial cell, and then he had grown it. Here in the tissue, we can have the tissue culture laboratory in the Department of Botany, and you can see here that with a small laboratory and with minor initiatives, we have started this tissue culture laboratory. So for the tissue culture laboratory, the nutrient medium is needed for the growth of the plant. Then aseptic condition should be maintained. Then controlled environmental conditions should be given for the growth and development of the plant. And then the observation and evaluation of culture should be there. Then recording of observation should be done. If it has been done properly, then we can do. Here we can see that the medium for the growth of, that is the MS medium, which has been formulated by Murashige and Skuda in 1962. And till date, even though n number of medias are there, but this is one of the basic medium which was given by Murashige and Skuga in 1962. Until date, we are using those medium for the growth and development. Then these growth hormones, which play a vital role in plant tissue culture, basically auxins and cytokinin. They are the two group of these hormones which works here. So among the auxins, indole acetic acid, indole butyric acid, nitric acid, naphthalene acetic acid, and 2,4-D can be utilized. And then cytokinins, carnitine is there, adenine is there, zeaxanthin and benzathine. These are they have been utilized. And then by their proper formulation, here in this check chart you can see that the action of growth hormones. At the left side you can see that cytokinin, whereas at the base the auxins are there, and here the formula is there. So if more part of cytokinin and less part of auxin is there, then shoot formation takes place. If more amount of auxin and less amount of cytokinin is there, then root formation takes place. And if both are there in equal quantity, then callus formation takes place. As you can see, the callus, you can see the callus here. So this is the callus of the Ceropegia bulbosa. So callus formation could be taken place with the proper with the help of then for giving the gelling property, then gelling agent like solidifying agent agar agar or clarigar or clarigel could be utilized. Then for in tissue culture, various steps are there. That is the preparation of explant, preparation of tissue culture medium, the sterilization of the explant, and then proper inoculation. Then this tissue culture is having several types. Here, this plant tissue culture has been divided into seed culture, meristem culture, protoplast culture, organ culture, bud culture, callus culture, cell culture, and embryo culture. And by taking these various explants, or explant means excise part of the plant, and they can utilize for the growth and development of the. Then after that, the proper inoculation should be there. Here in the during this inoculation, we have to maintain the aseptic condition, and we can do this operation in the laminar airflow. So this laminar airflow enables us for that. After the inoculation, then we can incubate this plant cell, and then after here after incubation, you can see that if they have been grown properly, then after incubation, if their cultures are growing, so we are go for the regeneration. So we do the subculture of this plant species and then grow them properly. So after subculture, in vitro culture is there, in which in vitro culture the plant cells are there, they have been maintained. Then so you can see that characteristics of plant cell or in vitro culture, several characteristics are there, in which environmental condition should be optimized for the growth on them, among which nutrition is there, light is there, and temperature is there. So ability to give rise to callus, embryo, or adult roots and shoots is able with the help of this growth hormones. Then with the help of this ability to grow as a single cell, so protoplast or microspores or suspension culture could be done. And then plant cells are totipotent, able to regenerate into whole. These are the various characteristics of the plant cell. Then there are three fundamental abilities of the plant, that is the totipotent. The plant cells are they are totipotent. They could be differentiated, and then they can have the competency. And by taking this advantage, we can grow these plants. So here the schematic representation is there, where explant is taken, and then it has been subculture on the medium. Growth is going to take place, and we can get the plant. So these are the important factors which enables for the growth. Growth media is there, environmental factors are there, and a source of explant. So by utilizing this, then here a certain number of growth hormones which help for the growth of the plant can be given, and their examples are shared. Here in this picture or in the slide, you can see that this with the help of this growth hormone, how one can mechanize, or how we can give the shape to the plant cells. Then various types of these cultures are there. Then seed culture may be there. As you know that we are sowing the seed in the soil and getting single plant, but with the help of this technique, with the tissue culture, if seed culture has been done, and we have added the cytokinins, then we can see that instead of a single plant, one can get n number of plants, and it is the example here that a single explant was taken, and you can see that n number of plants are they have been regenerated through the bottle. Then embryo culture is there, in which the embryo is there, it has been taken out, and then it has been cultured on the medium, and then from this plants are they there. Then among the organ culture, shoot culture is there, root culture is there, leaf culture is there, and flower culture. So among the shoot culture, shoot tip is already employed. In root culture could be done, which here we can call the rhizogenesis, or leaf culture is there, leaf can be done, or flower, among the flower ovary and anthers are they have been utilized for the culture. Here you can see the shoot apices there, it has been kept and then grown. Then here the here root culture there are rhizogenesis, and you can see that hairy root culture is one of the example, and the proper example is the growth of this Tagetes erecta. This roots of this Tagetes of Zendu can give the thiophils which has been utilized in the mosquito repellent coil, and then with that with the help of that we can do the rhizogenesis, and we extract the then ovary culture is there, and then anther culture. We know that this haploid plants is having a great demand in this laboratory, and with the help of that we are growing these plants with the here. Then callus culture is there. We know that callus is there, and this callus can also be in our laboratory. You can see at the tip it has got a pink color, means these cultures are there, they are responsible to give the flavonoids, and with the help of this callus culture you can do. And then cell suspension culture is there. We know that several cells are there, they can give the secondary metabolites, and this metabolites shall be utilized for the welfare of the human being. Apart from that, protoplast culture is there, or protoplast fusion could be facilitated. Then you can see the organogenesis. Here in the slide also you can see that the formation of organ could be done de novo. And then apart from that, one can do the micropropagation can be done, and then these plant cells are they have been utilized. Then here we can see that you can see in the slide that the micropropagation and in utero flowering could be seen. So this is the example of in our laboratory. This the cultures of this Celosia argentea, on which one of the students, Dr. Sachin Nirpani, has worked, and very nice. And we know that this plant Celosia argentea is utilized for the kidney stone, the seeds, and then we have to do the culture, and in utero flower of the flower formation was achieved. Then here here in the direct organizations, the proper example is the Solanum nigrum. So if the yes plants of Solanum nigrum, it may be leaf or it may be stem, if it has been kept on the tissue culture medium, then direct the somatic embryogenesis or organ formation takes place from the explant. Then somatic embryogenesis is one of the, we know that this somatic embryo, regeneration of embryos from the somatic cell can be called as somatic embryogenesis, and we have achieved that somatic embryogenesis in our laboratory. So this somatic embryos they are also important. Just I have told you that we can in root tumor formation can be taking place, and you can see in the slide you can see live in the culture that the root formation have been taken place with the help of this. So root formation has been occurred. And the roots are there. They have been employed for the betterment of the human being. Then what we are doing, we are not just ponder or what we are going to do, but how we are losing the diversity, and it is our the share for the conservation of the diversity. And for that conservation, what we are doing, that we can have our lion's share for the conservation of diversity. So it has been said that success in life ought to determine not only by accumulation but by contribution. And I am going to show our contribution. First of all, we have started our work on Dolichondra falcata, and it is one of the important plant. Dolichondra falcata, in Marathi we can call it as Medisingi, or in Sanskrit it has been called as Meshashrungi, which we prescribe in a herbal form for curing the piles. ज्याला मराठीमध्ये आपण मुळव्याध म्हणतो. So till date, more than 10,000 people has been cured. More than 10,000 people have taken the herbal medicine and they have been cured with these piles. Apart from that, we have the cultured Arva lanata, Celastrus paniculatus, Ceropegia bulbosa, Ceropegia lucia, and Ceropegia is going on. The citrus sinuses, one of our student Wagmare had worked on the citrus sinuses. Then another our student is had worked on Costus pictus. Mr. Or Dr. Avinash Jadhav has worked on this. Then here we can see the cultures of this Celosia argentina and Arva lanata. Then here, as I have told you, that the pigment formation takes place. In the slide at the left, you can see that the pigment formation, these are the flavonoids, and then which has been utilized for the betterment of the human being. These are the cultures of this Ceropegia bulbosa, and you can also see that then this is the anthurium, and just now Madam told that cryopreservation. So if we have prepared the synthetic seed, they could be cryopreservated and could be utilized in the here at the left the flowers you can see, and at the right slide you can see that the in vitro flowering in Ceropegia bulbosa. So this in vitro flowering could be achieved with the help of this plant tissue culture. So we have done that. Another plant we are working on, that is the Taca penetrifida, or one of the student, Ms. Swati, is working on this. At the right, you can see that these bulbs of this Taca penetrifida in China. This tacalin there it has been utilized. This Taca penetrifida oozes or gives rise to the cardiac glycosides, which has been utilized for the heart disorders. So we are utilizing so we are working on that plant. And then one of our students had worked on this Costus pictus or Costus speciosus. This has in Kerala medicine, it has been called as the insulin plant. And then we have successfully grown that plant. Another one of our students, Rupali Taur, Dr. Rupali Taur, had worked on this Cissus quadrangularis or Harjodi. So this plant is there, it has got activities on healing the wounds or bones. And then these fractures could be joined half of the time with the help of that. And this has also been utilized on in Marathi, it has been called a kestoda. So just the single this stem part is they have been churned and applied on the wound, and then it has been utilized for that sake. Then we have also worked on the banana cultures. Here these are the plant, that is the Dolichondra falcata. You can see the cultures, and then we are utilizing it for the sake of piles. Then here apart, you can see here another cultures of this Tylophora asthmatica. These cultures are they have been utilized for the welfare of the human being. Then here, these are the cultures of the Chiropygia bulbosa, the Costus pictus. Then here, one of our student, Rohit Shetty, has worked on the Solanum Xanthocarpum, Solanum virginianum. This plant is also local folk peoples are there. They are utilizing it for the healing of the piles. Apart from that, the leaf extract has been utilized for the regaining the hairs. So very much important plants are there. One of our student, you can see here the Celosia cultures, you can see nicely, and these Celosia seeds are there. They have been utilized to cure the kidney stone. And here you can see that the nice cultures are there. They have been growing. Then these are the Tylophora again cultures. You can see this is the cultures of Celestial Panicula, as it is a woody climber. But we are succeeded to grow this. One of our student Megha Sheval is working on this plant. Then these are the cultures of Catharanthus rugius, white, and then as we know that this the Catharanthus rugius uses the vincristine or vinblastine, and then it is an anti-cancer drug. Here again we are going for the conservation, and here you can see that the cultures of the Terminal Arjuna. One of the student is working on this Terminal Arjuna, which is one of the important plant. We can have the Podocarpus growing in our the laboratory, botanical garden, and we have tried to culture this Podocarpus. Then these are the cultures of Solanum nigrum. These are the cultures of Withania somnifera, which uses the withanine, and then you can see the nice cultures are there. Then in our department, we can have this Professor Pokle run a project of worth 65 lakh rupees on the Abutin and Ranadi. So it is one of the rare and endangered plant, and we have cultured that in our laboratory. Then Citrus sinensis, as it has got the dieback in this plant, we have worked on that, and we have seen that if we have these viruses are there, they are responsible for dieback, and then we can if this cultures has been utilized for the growth of the clones, and then no dieback disease could be found. These are the Datura metal. This is also one of the important medicinal plant. Then here the Gloriosa superba, in Marathi it has been called as Kallavi, and these plants are there. They have been grown in our one of our student Jyoti Godke is working on this Gloriosa superba. In the ancient times, the roots of this tubers of this plant was utilized for the paste was applied on the during the labor pain, and the normal delivery was going to take place. So we are also growing, and you can see the cultures are there in which in vitro rhizogenesis has been taken place. Then here one of our student Pranita Zamdade has worked on Agile marmelos. In the Shravan or Shivratri, we know that the bale or Agile marmelos is there. It has been utilized, and Lord Mahadeva, as we praise him that Karachana Kutamva Kayajam Karmajamva Shravana Nayana Java Man Sama Paradam Ritam Sarvame Takshamaswa Jai Jai Karunabhe Shri Mahadev Shambhu. And with this pray, we are going to offer this below patra to this Lord Mahadeva. And we are in these days, you can see that in front of each temple or Mahadev temple, you can see that how the hundreds of people are there. They are selling this below patra, and due to the huge cutting of this trees, no fruit and flower formation going to take place. And you can see that this is one of the important medicinal plant, and then it has come into endangered due to the so we have tried. It is hard to culture this, but we have done that cultures, and you can see that nice cultures are there. They are growing. Then one of our students is working. We know that Taman, the Lagestromia speciosa. So you can see here that in vitro this embryogenesis has been taken place, and we have grown this Lagestromia as well. Then we frequently visit the localities of Melghat Lake, and just from that Melghat forest, we have collected this Taka Venatipidaris Levante petolite. One of our students is working, and you can see the live culture of this Taka Penatipita, the live, and you can see that you need to do a formation. So they are collected from that. And in last year, we have visited that locality. And among the big jungle, only some part is there where the tiger resides. And in that locality, this plant is there, it grows. So we have collected that plant. And then here you can see that in utero tuber formation is that it has been seen. And then we have done it properly. Then we are also try to culture the Melia azariacta or bakan. This is also one of the important medicinal plant and utilized as a fodder crop. Then banana, we know that several laboratories are there, they are working on this banana. So we have also cultured this banana. Then herbal anatomy, already you have seen the cultures. Then these are the slides which shows that various the plant which has been cultured in our laboratory. And then this is the ground zero report where on the field this work was whenever I was there in the Satara, Yeshwantara Institute of Satara. So ND Patil Sahib, one of the greatest reformer, he had appreciated our work. You can see that students are there in your Earn and Learn scheme. They are working on banana tissue culture. And at their back, you can see that these cultures of bananas are there. Then Professor Bhoite sir, he was the secretary of this Rayat Shiksha Sanstha. Here, in Rayat Shiksha Sanstha, Budharja Mulek, one of the social reformer, is going to see the cultures. Then these are my inspirations in the university. The Kotapalli sir has given me an opportunity. My teachers are there. It may be Vishwa Prakash sir may be there. In Maulana Azad College, my teacher Mogarkar sir and Sabera madam is there. And apart from that, in the right, you can see that Professor Kavi Kishore sir from the Usman University, Hyderabad, Department of Genetics, always inspires us to do this. And he is working, giving us inspiration and helping us in several ways. Then this tissue culture is not only in the culture in the laboratory, but we have spread it throughout the Marathwada, parts and corners of Maharashtra. And we always, they organize the workshops for the students. You can, in the middle, you can see that during the tissue culture workshop, and at the right, you can see that several students do visit in our tissue culture laboratory, and they are going to take the benefit and how the culture is doing. And here, these are the visitors of tissue culture laboratory, routine visitors. And then you can see that the workshops are there, they have been organized in government of college of science. And then our students are also, they are going to train them. This is the seminar which has been taken place in Partur, Lal Bahadur Shastri Mahavidyalaya Partur, and a huge response was there. More than 300 to 400 participants have taken participation. This is the workshop which has been taken place at the Vasant Mahavidyalaya case, and Professor Kashit sir had taken initiative, and the students have taken benefited of this tissue culture workshop. Then not only students but college students are there, they are going to take benefit, and we always give the demonstration to the people also. This is my team, and during this Vice Chancellor Chopre sir, a huge amount of currency was given to run our tissue culture laboratory, and it is our pride that this former governor Shankar Nayan has visited our laboratory and he inspired us. So this is the these are the publications. This is the list of students, and they have worked on various aspects. Till that, 12 students have completed their PhD, and 2 students have submitted. And these are the newsroom or the cognizance which has been taken. These are the sure workshop highlighted. Then such works are going on for the cause of the nation. And then you can see that not only a BSc PhD student but our MSc student have taken 2 prizes in workshop in Milin College, which has been yesterday, and then the 2 prizes they have won. So MSc students, as a part of their this project, they are working on this, and then we can getting the results. And then for the conservation of this, Lord Shivaji or Shivarayani Ajnapatra काढलं होतं, and he tells us that the wood which has been utilized for the various purposes should be taken with the permission of the that the grower then you should not be cut or you should be done every year. And lastly, he says that these plants are they are important for us, so we have to conserve this plant. And lastly, with the help of this plant, it has, as we say that that Lord given us a various opportunity, and then so we have to conserve this plant. And lastly, I praise to the Lord that na toham kamay rajam, na swargam na punarbhavam, kamay dukha tapnanam prani namarthi nasham. Oh Lord, you have given me such a big opportunity to serve the nation or serve the society. Till the as I work, I should be this work should be for the cause of the noble cause, should be there for the welfare of the human being. And lastly, one of the very nice poem is there, and that poet said that हे मानवा कृपा करून विसरू नको, हिवाळ्यात तुझ्या शेकोटीचा ऊब मी आहे, उन्हाळ्यात तुझ्या डोक्यावरील छाया मी आहे, माझी फळे प्रवासात तुझा भूक भागवतात आणि त्यांचा रस तुझा सुकलेला घसावला करतात. तुझ्या घराचं मी खांब आहे, तुझ्या छपराची तुळे आहे, नदी नाळ्यातून येणारे तुझे नाव मी आहे, विश्रांती देणारा तुझा मंचक मी आहे, तुझ्या कुंदरीचा दांडा आहे, नांगराचा फाळ आहे, गाडीचं चाक आहे, पाळण्यामध्ये तुला जोजवणारा मीच होतो आणि स्वर्गाच्या स्वारीचेही रथ मीच असणार आहे. तर हे मानवा कृपा करून विसरू नको. तर स्मृतीचा हा जिवंत झरा असाच वाहत राहू. So this knowledge of the plant tissue culture should be continuously goes for the cause of the nation and the welfare of the human being. So with these words, I am very much thankful for giving me an opportunity, and it is my appeal to all that if anybody कुणालाही जर टिश्यू कल्चर लॅब जर डेव्हलप करायची असली तर नक्कीच त्यांना आपण मार्गदर्शन करूया. कारण की एक जरी कॅलस जरी होत असलं किंवा एक्सप्लांट जरी वाढत असलं तरी मुलांना त्याचं कुतूहल वाढत असतं. आणि सध्याची आपल्याला माहिती आहे की कंडिशन पाहतो सिनारियो की वतन का जो हाल सुनेंगे तो पत्थर भी आंसू करेंगे, अरे खो गई हे भीड़ में आदमी जिससे ढूंढने जमाने लगेंगे. तर ह्या काळामध्ये विद्यार्थ्यांचं वाली आपणच आहोत, त्यांना कशा प्रकारे फुलफिल करायचं हे फक्त आपल्या हातात आहे. आणि जितकं आपण त्यांना ज्ञान नॉलेज देऊ तितकं वाढतच राहणार आहे. तर ते आपण देत राहूया आणि हे समाजाची सेवा करू द्या. तर ऑर्गनायझरनी मी शतशः आभारी आहे. तिलावत अली साहेब यांना ट्रिब्यूट देतोय. तुमचे कबीर सर आहेत, प्राचार्य कबीर सर, प्रोफेसर मिलिंद जाधव सर असतील, त्यांनी मला ऑपॉर्च्युनिटी दिली ऑर्गनायझर्स. आणि ऑडियन्समध्ये माळी सरांना सुद्धा पाहतोय, प्राचार्य माळी सरांना मी पाहतोय. आय एम थँकफुल टू यू फॉर पेशन्स लिसनिंग ऑफ दिस लेक्चर. व्हेरी थँक्यू. ओव्हर टू यू जाधव सर.
Thank you, sir. Pandure sir, surely we send our students to you for the tissue culture program. And today's in our Pandure sir's lecture, sir has highlighted on the importance of the biodiversity and its type of biodiversity. He has explained the importance of each and every plant. Each and every plant is useful to us, and so that we have to conserve our biodiversity. For that we have to improve the method of plants by tissue culture process. And in that tissue culture, sir is the master of the tissue culture. Thank you, sir. Thank you lot. And we are grateful to you for your valuable guidance. Thanks once again. We have to protect the environment to protect ourselves. So let's come together and save the environment. Dear participants, now we are proceeding to the next session. It is an honor for me to introduce and welcome to the resource person of our next session, that is Dr. Vasant Mali sir. Dr. Vasant Mali sir is working as a principal and at J. Vatumal Sadhubera Girls College, Ulhasnagar. His area of research is aerobiology, taxonomy, and mycology. Under his able guidance, 3 students completed MPhil and 2 students completed PhD, and 5 students are working for PhD. Sir has published 40 research papers in national and international journals. Dr. Mali has worked on different professional and academic bodies as an organizer, convener, chairperson, and member. Sir is awarded by Vriksha Mitra Puraskar, Shikshak Ratna Puraskar, and Fellow of ISST. Dr. Mali has written 3 books. Sir has visited 13 countries such as Nepal, Bhutan, Bali, Indonesia, Thailand, etc. Dr. Mali is renowned as academician, researcher, and administrator. Now I request Dr. Mali sir to deliver this talk. Dr. Mali sir, please.
Hello. Hello. I am audible?
Yes.
Sir, please give me screen share.
Yes, sir. Yes. मुझे screen
Yes, sir. Yes, sir.
Yes, sir.
Okay.
Yes, sir.
सर शेअर करा सर.
हाँ करतो सर 1 मिनिट.
येस येस येस. बरोबर आहे. हा.
आता thank you म्हणे.
हॅलो.
हा सर शेअरिंग करा ना.
शेअरिंग नाही झाली काय? शो नॉट स्क्रीन.
Yes, yes.
नाही ना अजून शेअरिंग झालं नाही ना. शेअर करा ना.
1 मिनिट.
स्टार्ट.
सर 1 मिनिट.
नो प्रॉब्लेम. काय अडचण नाही.
Take your own time.
करू का? झालं.
सर स्क्रीन शेअर झाली?
येस झाली सर. दिसतंय. नो प्रॉब्लेम.
Okay, sir.
Go ahead.
Okay.
Go ahead.
Yes,
Go
sir.
ahead. No problem.
Yes, sir. Yes, sir. Good afternoon, everybody. I am very much thankful to management of Sir Syed College, Aurangabad, organizing secretary, Sheikh Ahmad sir, convener Professor Dr. Milind Jadhav, co-convener Ahuja madam, Sutar madam, given me opportunity as a resource person and sharing my view in front of you. And today's eminent person in this conference, ex-BSUD director, my friend Dr. Anil Patil sir, Pandure sir, and Meenakshi Mahajan sir. My today's topic is an introduction to common wood rot fungi in tropical forest. I am also basically from aerobiologist, but from 10 years, I am working in Apollophorales and wood rotting fungi. My 5 students are also working in these groups. I am only given in shortly material and methods. Collection of wood rotting fungi specimens was collected from tropical forest. Collection was done every year during the period from July to August and September to middle October, because that time the period was for the found to be congenial luxuriant growth, development, and availability of Apollophorales and wood rotting fungi. Fruiting bodies of Hymenocrates were collected from stem and old trees in forest, roadside, old stem, of timbers, and also some wood rotting fungi are also collected from the living plants. All the collected specimen were properly sun and air dried, then kept in brown bags and their top tightened with rubber bands as per the International Herbarium Guideline. Each collected specimen was labeled with its name, locality, and host name. 1 or 2 naphthalene balls kept in each pocket to avoid damage by insects. Then laboratory work, take a thin section of the context with the sharp blades. Then these sections observe under the microscope for the identification. Then wood rotting fungi represent in an artificial assemblage those fungi which are sporulating over living dead twigs, branches, trunks, stems, and fallen logs with morphological adaptation. They play pathogenic role by destroying valuable forest. But important point, keeping the biomass balanced ecologically, white rot and brown rot. Basically, wood rotting fungi are identified on the basis of morphological as well as anatomical character. Anatomical character, the shape and size and color of spores. Then types of hyphae, mostly 3 types of hyphae: generative hyphae, skeletal hyphae, and binding hyphae. Then another characteristic identification identification on the basis of basidia, cystidia, etc. Then brown rot. Brown rot causes the wood to crack in a cuboidal manner. The wood also goes slightly darker in color. Then cubes may vary in size depending on wood and conditions, but they are generally very visible and distinct, especially as wood dries. There are many brown rots, dry rots, for example, Serpula being just one. Thus, if the rot is identified as one of the brown rot, and then it could be dry. Then white rot. White rot attacked by look completely different to attack by brown rot. The wood takes on a fibrous appearance and tends to go slightly lighter in color. This may not be too noticeable. There is no cuboidal cracking as in the brown rot. That tends to be more common in hardwood. Hello.
Yes, sir.
Disturbance hota. Okay, sir. If the damage is shown to be from one another white rots, then it is certain that the rot is not dry rot. Then I have given some photographs of the wood rotting fungi. Suppose any queries, please call me on my mobile number or send your queries on my email address, because I am shortly describe here. First, Amyloporus campbelli. These all 3 photographs are in habit. Stiff basidiocarp stipitate, strongly decurrent pores, dominant generative hyphae. In the next, both simple, septate and clamp generative hyphae. Presence of glioporous hyphae and equinulate, amyloid and Cyanophilus basidiospores. Then another photographs is Trimetes villosa. Then Cellularia acuta in first photographs. This is a habit in the forest. Then second also habit in third photographs. In third photographs, comparative, this is a pilear surface and this is a hymeneal surface. And in photographs, last photograph 4th, the hymeneal surface.
सर फोटोग्राफ शेअर स्लाइड शेअर होत नाहीये तर
स्लाइड शेअर होत नाही सर.
नाही पहिली स्लाइड सुरू आहे.
एक मिनिट.
स्क्रीन हँग झाला का?
नाही.
हो ना, इथं काय होतंय, पहिलंच ट्रान्समिशन दिसतंय आम्हाला.
आता दिसले?
नाही.
आता दिसतंय सर?
नो सर.
नो. एक मिनिट.
आता शेअर करा सर.
हो. दिसते?
हा आता दिसतंय सर.
Okay sir.
Okay.
This is a next photograph is a Cellularia acuta. Cellularia first photographs is in a forest. Second photographs are also forest. Third photographs two comparatively two specimens. First specimen is the pilear surface. This is a morphological characters, or it is also identify on these morphological characters. And second photographs is comparatively hymenophore, and these hymenophores are enlarged in last photograph. Basidiocarp is annual. Its life cycle completed within 1 year or short-lived. Pileate to effuse reflex, sessile, usually imbricate, hypal system, trimatic, generative hyphae, hyaline, binding hyphae, tortoise. It is a characteristic of this Cellularia acuta. Then Inonotus ricci. Inonotus ricci in a two stages. First stage is globular in form, and chlamydospores are very abundant in this stage. These specimens are collected from Aurangabad bus stand. Aurangabad is a bus stand where the bus is live. The bus stand, then letters left side Dalbergia sisal plants, and these plants are after 1 year they are totally dry and fall. Basidiocarp's annual, sessile or single, imbricate, separable, aplanate to angulate. Hypal system monometic, only one type of hyphae. This is generative hyphae. Thin to moderately thick-walled, spores abundant, thick-walled, brownish, reddish brown in KOH. It is also another characters. Spores ellipsoid to ovoid, chlamydospores abundant in the context tissues. Irregularly in shape, globose to ellipsoid. Then next is a Flavodon flavus. Flavodon flavus is very common in a tropical region. They are mostly found on dead stems in photograph first, and also found on living plants on the trunks. In photographs first, the specimens flavodons are grow on the living stems. Then in second photographs, and in third photographs, this pilear surface and this is a hymeneal surface. Also in these photographs, they are comparative pilear surface and hymeneal surface, and this is a context. Then Basidiocarp's annual, this first in starting they are in growing. This is a rhizopinnate forms, and then it is a effuse reflex. Basidio of reddish to brown in KOH, a rhizopinnate or pileate. Hypal system diametic. Two types of hyphae, generative hyphae, hyaline, and the simple septa. Then second, skeletal hyphae also hyaline but thick wall. Spores broadly. Then next is a Ganoderma applanatum. Ganoderma applanatum are mostly found on the living stem trunk. Basidiocarp perennial, sessile, woody to corky, applanate, hypal system trimatic, generative hyphae inconspicuous, thin wall with clamp, skeletal hyphae thick walled, brown septate, binding septate, binding hyphae septate, spores ovoid and truncate at the distal end. By tunicate, exposure hyaline. In first photographs shown the basidiocarp growing on the living plants, and this is a pilear surface. Then next is a Ganoderma lucidum. Ganoderma lucidum is very common. Mostly they are found at the base of the trunk, at the base of the dead stem. Ganoderma basidiocarps annual, laterally to centrally stipitate, pileus, reniform or dimediate, or when they are in a young conditions, after the brushing, its color is a change. Hypal system trimatic, spores ovoid, or its main characteristic. This Ganoderma are number of species. They are classified on the basis of cuticles and spores. The truncate spore is a main characteristic of the Ganoderma. Then Phalinus badius. Phalinus badius is very common in Marathwada region, Nanded and Parvani. This is a dry region. In this dry region, the dominant of vegetation of Acacia nilotica, and these specimens are mostly found on the Acacia nilotica. Rarely they are found on Peltophorum pterocarpus. Basidiocarp perennial, sessile, hoop shape to ungulate to appearing somewhat pendant, and these specimens are very easily detachable from the host. Hypal system diametic, generative hyphae hyaline to pale yellow, skeletal hyphae thick walled, hymeneal setae absent or very rarely present. The main characteristic of the Phalinus is setae are present. It is a main characteristic. In older space, very rarely present in older species. Spores are broadly ellipsoid. Then next is a Pycnoporus syngenous. Pycnoporus syngenous only two species, Pycnoporus cymbarinus and Pycnoporus syngenous. Pycnoporus syngenous are separated from the Pycnoporus barinus. Basidiocarp annual, sessile to effuse reflex, single or imbricate, cluster, dimediate. These are mostly, these Pycnoporus are mostly found on dead logs. And Basidiocarps orange colored, hypal system trimatic, pore surface dark range red orange buff, spore cylindric, slightly curved, hyaline, and smooth. Then next is Arleria scabrosa. Arleria scabrosa are not dominant in Marathwada. They are rarely found. Basidio ka first photographs is on the dead wood, this number of specimens. Then in second photographs, this is a taken photograph from lower side. This is a hymeneal surface, and these hymeneal surface are extended in these photographs. Basidiocarps annual to periennial, effuse reflex, hypal system, trimatic, three types of hypal systems. Spores hyaline, cylindrical, oblong, or ellipsoids. Then Phaeolus grammocephalus. Phaeolus grammocephalus in first photographs. This is a imbricate manner, and number of Phaeolus on the dead wood. They are mostly found on Ziziphus jujuba. Then in last photographs, they are compared. This is a hymeneal surface. Sorry, this is a pilear surface, and this is a hymeneal surface, and this is a context. Basidiocarp annual, thin, soft, bristle on drying, dimediate, flabelliform to spatulate, laterally stipated. Then Basidiocarps thin, soft, bristle on drying, dimediate, flabelliform to spatulate, laterally stipated. Hypal system, diametric hypal system. Only two types of hypal systems: spore cylindrical, hyaline, and smooth. Then Grammotheli fuligo. Grammotheli fuligo are mostly found on monocotyledonous plants. Found on monocotyledonous plants. Then first photographs, this on the branches of monocotyledonous plants. Then also on the base of the leaf scar base. Basidiocarp annual, rejuvenate, just paste-like, pores surface cream colored to bluish gray to pale umber brown, darkening with age, pores regular or irregularly epicoid, becoming labyrinthine bluish gray to pale umber brown. This is a hymeneal surface, and this is a pores. Then Dendrophidia present. Dendrophidia, it is a identification characters of Grammotheli fuligo. Then Hexagonia tenuis. Hexagonia is differentiated into 4 species, but in Marathwada or in a tropical region, mostly found Hexagonia tenuis. The poroid surface is a hexagonal in shape, and it is a main identified characters of identification of the Hexagonia tenuis. Hexagonia are mostly found on the mango tree in a tropical region, Basidiocarps annual to periennial. Pore surface small, brown to milky, coffee colored, often with a grayish to ash bluish tint. Pore angular or hexagonal, they are shown in last figures. Hypal system, trimatic, spores cylindrical, hyaline, and thick walled. Then next is a Microsporus xanthoporus. Microsporus xanthoporus. They are also found, mostly found in a tropical forest on, especially on Terminalia chigula and Terminalia bellarica. This is a pilear surface, and in last photographs, they are shown pilear surface as well as hymeneal surface. Basidiocarps annual, solitary or in small cluster, centrally or laterally stipated, and usually in fundibuliforms. coriaceous, when fresh. They are when fresh, this color is also change, and after dry also change its color. And when fresh, this is a fleshy, and when dry, it is a hard. Glabrous to hirsute and zonate. These zones are present on the pilear surface. Pore surface, cream to pale buff, almost pure white, toward the margin of hypal, towards the margin. In these photographs, they are white towards the margin. Hypal system trimatic, and then strongly corolide, dichophytic elements often present along the discipient. Then I am only shown another only photographs, not given the detailed characters. Cyathus striatus, this is also another wood rotting fungi. Then Funalia caperata, then Funalia caperata, Inonotus pachifolius. Inonotus ricci are very commonly in a tropical forest, but Inonotus pachifolius is not a dominant. Then next, Trimates apiara. Trimates are 4 to 5 species are found in the tropical region. In first photographs shown the pilear surface. In second photographs shown the hymeneal surface. Then next, Leotrimates lactania. In first photographs, this is a pilear surface, this is a hymeneal surface, and this Leotrimates on the host. Then in next is a Trimates leonina. Trimates leonina are separated from the Funalia leonina. Its first name is Funalia leonina, and they are separated from the Funalia, and they are included in Trimates. Leonina species is same, but only a genus is changed. Basidiocarps annual. These Trimates leonina are mostly found in tropical region, especially on the Mangifera indica. Basidiocarp annual, pileate, soft when fresh, tough when dry. Then pileus flat to convex, covered with dense mat of strigose hairs, and it is a main characteristic and character of identification of this species. Becoming hispid in old specimen, pure white when fresh, soon straw colored, and finally grayish in old specimen. A common wound, parasite on dead deciduous wood, particularly in Mangifera indica. The pure white fruiting bodies with dense mat strigose hairs on pilear surface, distinguished with this species in the field very easily. Then next is a gynoduenting, then Serpula similis, then Podosepia, then Duportella tristicula, Heatherstromia frigici, then Hymenocete concata, these members from Hymenocetaceae, and the Hymenocetaceae is a very large family. Then another from another group, Hypoxylon, Hemistroma, then Xylaria symplosi, then Xylaria multiflex, then Dacropinax spatularia, and Trimella fusiformis, Gymnopallius, then Lantinus sajorcaju, then Schizophyllum communi. Schizophylum communis are mostly found on all host or on plants, and Schizophylum communi, it is only single species in genus Schizophylum, but it is a variously colored, brown color or mostly white color, but only single species in India or in world at that time, Schizophylum. Then Trogia infundibuliformis. And lastly, all photographs are mixed, and this photographs is a Inodontus ricci is in a first stage. Okay, thank you. I am very much thankful to management and Dr. Milind Jadhav sir. Thank you, sir.
Sir. Thank you, Mali sir. Mali sir has introduced to common wood rot fungi in tropical forest nicely. The lecture is very useful to the researcher who want to study the fungi. Thank you very much, sir, for your informative presentation. Sir, we are grateful to you for your valuable guidance. Thank you so much, sir. Dear participants, now we are proceeding to our important part of the e-seminar. Now I request Dr. Milind Jadhav sir to proceed for the valedictory function. Dr. Milind Jadhav sir. Dr. Milind Jadhav sir.
व्हिडिओ पण ऑफ केलाय आणि ऑडिओ पण ऑफ केलाय. छान लेक्चर झालंय. कमी वेळेत मी एक 30-35 मिनिटं घेतलेले होते.
मला कामाला नाही वाटत आता व्हिडिओ ऑडिओ
आज़्म मुस्तहकम हो तो होती है बलाएं पसपा, आज़्म मुस्तहकम हो तो होती है बलाएं पसपा, कितनी मौजों को पलट देता है साहिल तन्हा। Honorable Chief Guest, Dr. Meenakshi Mahajan, Associate Professor, Department of Botany, Fergusson College, Pune. The guest of honor, Dr. Shamama Parveen, President, Rehbar Educational, Cultural and Welfare Society. Other dignitaries and the participants. We are at the concluding part of the seminar. By the grace of Almighty, this one-day national seminar on recent trends in plant sciences witnessed a huge response. Despite being Sunday today, the participants remained actively involved throughout the day. We surely bestow our gratitude to all the resource persons and the participants. I invite the convener of the seminar, Dr. Milind Jadhav, to present the seminar report. Dr. Milind Jadhav.
Audio. Good evening, one and all. Now, dear participants, we are at the end of this today's seminar, that is a valedictory function. To this valedictory function, I welcome our chairperson, Dr. Shamama Parveen ma'am, principal, Dr. Sheikh Kabir Ahmed sir, today's valedictory chief guest, Dr. Meenakshi Mahajan from Fergusson College, Pune, Professor Narayan Pandure, Department of Botany, Dr. Babasaheb Ambedkar Maratha University, Aurangabad, Principal Vasant Mali, J. Batumal, Girls College, Ulhasnagar, and all my researchers, teachers, and students. In morning session, today's e-seminar was inaugurated by the eminent personality, administrator, and researcher, Professor Surendra Singh from Department of Biological Sciences, Rani Durgavati Vishwavidyalaya. To this inauguration session, the chief guest was Dr. Shamama Parveen ma'am, Dr. Sheikh Kabir sir, and guest of honor, Professor Shailesh Jadhav sir from School of Studies in Biotechnology, Pandit Ravi Shankar Shukla University, Chhattisgarh, Raipur, Chhattisgarh. The inauguration session was followed by a keynote address, which is delivered by Professor Surendra Singh. The lecture was delivered on the topic, that is algae and human welfare. In his lecture, Professor Surendra Singh nicely spoke on algal biotechnology. He mainly focused on different aspects of algal biotechnology, such as food and feed from algae, then role of algae in therapeutics, role of algae in biofertilizer, extraction of biodiesel from algae, production of fine chemicals from algae. Overall, the lecture was very nice and valuable, informative, and knowledgeable. This lecture will be very much useful to the upcoming researchers who want to work in the field of biotechnology. Now, the keynote address was followed by the invited lectures of Professor K.K. Sahu sir from Pandit Ravi Shankar Shukla University, Chhattisgarh, Raipur, Chhattisgarh. Professor Sahu sir has mainly focused on toxicity of metals, effect of toxicity of metals on various crops, alteration, morphological, physiological, and genomic alterations by the toxic metals that has been well documented and that has been presented in good manner by the Professor K.K. Sahu sir. Professor K.K. Sahu sir also briefly described, also explained a methodology for the study of metal toxicity on different plants, that is on the genomic level. The lecture of Professor Sahu sir followed by a lecture of Professor Meenakshi Mahajan, who is working as an associate professor in Ferguson College, Pune. Ma'am delivered a lecture on role of biotechnology for the conservation of biodiversity. Madam have explained a number of methods for the conservation of biodiversity, mainly ex situ and in situ conservations, cryopreservations, like that, tissue like that. Then in next lecture, in next invited talk, Professor Narayan Pandure from Department of Botany, Dr. Babasaheb Gunata University. Dr. Pandure sir mainly focused on conservation of plant species through a tissue culture technique. Professor Narayan Pandure sir is one of the important authority in tissue culture of Marathwada University. He and his team extensively worked on and working on tissue culture to conserve the endangered plants of Marathwada region. And in last lecture, last but not the least lecture, Professor Mali sir from a J. Batumal College, Professor and Principal Mali sir from J. Batumal College, Ulhasnagar, he mainly delivered a lecture on, the lecture was very basic, having a basic concepts regarding the mycology, but that is very important for a beginners who wants to work in a mycology, especially wood rotting fungi. Now, this lecture was very beautiful with a nice presentation. Dr. Mali sir has extensively worked on a wood rotting fungi in Marathwada region. He is the authority in wood rotting fungi as far as the Marathwada region is concerned. His lecture was so nice and knowledgeable and very informative. Dear participants, for this, our e-seminar, 223 participants have registered and participated in this seminar. The participants from Andhra Pradesh, Telangana, Haryana, West Bengal, Tamil Nadu, Assam, Rajasthan, Uttar Pradesh, Madhya Pradesh, Chhattisgarh, Pondicherry, Gujarat, and from Maharashtra, the participants were participated actively. I am very much thankful to the our college chairperson, Dr. Shamama Parveen ma'am, and our principal, Dr. Sheikh Kabir sir, who has encouraged us to organize this e-seminar, which is very important, which is very useful to the researchers, teachers, students, academicians. With these few words, I request Dr. Azhar sir for a further proceeding. Thank you.
Thank you very much, sir. A good deed is one that brings a smile of joy to the face of another. Some of the participants are interested in sharing their views on this occasion. Let us listen to them. Firstly, I invite Dr. Jagdish Saudekar from Yashwant Rao College, Sillor. Dr. Jagdish Saudekar, sir.
Hi, na?
Sir, are you there?
Yes, sir.
Please share your views.
Yes, yes.
Good evening to one and all. Today, Sir Syed College Aurangabad organized one-day national e-seminar on the theme Recent Trends in Plant Sciences. So, this theme, it was too much useful for faculties, researchers, students, academicians of the plant science field. So in first session, keynote address was given by Professor Surendra Singh, in which he focused and nicely presented a deeply studied topic of algal biotechnology, aspects of algae and its scope in various fields. Thereafter, we got a fruitful knowledge, a deep information of toxicity of metals on crops from Professor K.K. Sahu. Thereafter, Professor Narayan Pandure sir focus on tissue culture and biotechnology in deep way. Then Professor Meenakshi Mahajan ma'am focus on role of biotechnology on conservation of biodiversity. Thereafter, we got fruitful information from Principal Mari sir on wood rotten fungi. So it was nothing but a definitely successful national e-seminar. So I congratulate to management of Sir Syed College, principal, then respected convener, Professor Jadhav sir, Professor Sutar ma'am, Ahuja ma'am, and all team of Sir Syed College. So I have expressed my sincere thanks because principal sir and convener gave me a chance to express my views regarding this conference. Thank you, sir.
Thank you very much, sir, for your encouragement. May I now invite Shama Rajmani from Barwale College, Jalna. Shama Rajmani.
माइक चालू आहे तुमचं इथं.
तुमचं माईक
स्पीकर एक बंद करा.
सर क्षमा करो ना स्क्रीन.
क्षमा राजमानी माहिती नाही.
क्षमा.
Yes.
Yes.
I'm here.
Yes, there. Yes, yes. Rajmani madam, please continue.
Okay. Thank you, Dr. Milind sir. Nice program organized by you, all the committee, organizing committee. I started my work from attending your program from inauguration function, sir. And I like very much the starting program by your college management. And then Professor Surendra Singh deeply given a lecture who educate us on the algal biotechnology, especially on the spirulina, which is a high protein algae. And I like so much this talk, sir. And then Professor K.K. Sahu from Raipur University, he is also delivered very nice lecture, and he in toxicity of such metals is very nice. Dr. Meenakshi Mahajan ma'am from the Ferguson College, a very nice lecture on technology in the conservation of the plant species. Here in she had given very nice technique of seed bank conservation and in situ and ex situ methods, deeply elaborated by her. And then I also like in this talk the detailing about the cryopreservation with different steps, near about 7 steps she had explained. This is very informative, educative talk, sir. After that, Professor Narayan Pandure had also given nice explanation about the tissue culture method by using different hormones like indo-lactic acid, biotin, zeatin, and all, 2,4-D for the root and shoot growth and callus growth also.
Very nice, sir. And like this, Dr. Vasant Mali also had given very nice lecture on the wood rotting. These are the 5 different color talk will be there. I enjoyed too much today. Thank you so much. Dr. Sangeeta Sutar, Dr. Sangeeta Ahuja, thank you so much. It is nice organizing program. Thank you, sir.
Thanks a lot, madam. We are quite fortunate to have Dr. Meenakshi Mahajan from Ferguson College, Pune, as the esteemed chief guest. After listening to her beautiful talk, we are here again to request her to enlighten us on this auspicious occasion. Dr. Meenakshi Mahajan, ma'am.
Thank you so much. Can you hear me?
Yes, yes.
Good evening, one and all. At the outset, I am thankful to Principal Dr. Ahmed, Professor Dr. Milind Jadhav, Head and Convener, Department of Botany, who have given me this honor to be the chief guest in this seminar. Respected dignitaries on the dais, Dr. Shamama Parvin, President, RECWS, Guest of Honor, Dr. Sheikh Kabir Ahmed, Principal, Sir Syed College of Arts, Commerce and Science, Professor Dr. Milind Jadhav, Head and Convener of Department of Botany, Dr. Sangeeta Ahuja, Co-Convener, Dr. Sangeeta Sutar, Coordinator, other members of Organizing Committee, all invited speakers and fellow participants from different college and all dear students. I want to congratulate management principal Dr. Ahmed and head of the department, Dr.
Madam,
Yes.
Excuse me, madam.
Yes, yes.
Video on करा.
Video on. Yeah.
Yes.
Yes, now it is on, sir. I want to congratulate management, Principal Dr. Ahmed and Professor Dr. Milind Jadhav for hosting a wonderful and stimulating national seminar. It's a fruitful and amazing time for all academicians, researchers and students to utilize time in pandemic situation. Invited talks of this seminar will definitely create some curiosity in the mind of students and researchers about higher and lower plant diversity, their taxonomy, and their conservation. Albert Einstein said, curiosity is more important than knowledge because curiosity leads to learn more. If science has to grow, the younger generation who are the future of the country should take up the challenge. COVID-19 pandemic forced to shut down all the educational institutes across the country, but we cannot stop our learning and sharing our knowledge. In this crucial time of COVID-19 pandemic, the exposure to online e-seminar with the aid of internet technology is indeed a blessing to the teachers, academicians, students, and researchers across the nation. In today's seminar, all of us came to know about importance of lower plants in human life. Lower plants, they are used as non-conventional food, and to fulfill all the demands of basic demands of a human being, it's a need of time to give more concentration on the non-conventional use of lower plants. So from that point of view, we got very good information, how algae they are used as food, what is the role of algae as biofertilizers, what is the use of algae as biodiesel. Besides this, we came to know about the role of heavy metals, how the lower plants, they play an important role in during the heavy metals one. Then we are much clear with the conservation aspects of the plant species by using biotechnology, and how we can conserve them by using tissue culture mechanism, tissue culture technique. So besides this, we came to know about the identification of wood rotting fungi. So it's a basic taxonomy, which is the backbone of all sciences. So Mali sir, he has conducted his lecture on the identification of wood rotting fungi, which will be very much helpful for the students in identification. So with this few words, I conclude my speech. Thank you so much.
Thanks a lot, Meenakshi ma'am. We shall remember your keywords. We shall remember your guidance. May God bless you for all the things. We are here with some important instructions for the participants. All the participants are required to fill the online feedback form, which will be sent to you immediately after the valedictory session is over. And all are going to get e-certificate. For getting e-certificate, the participants require 3 days. So by the end of 3 days, all the participants are supposed to get e-certificates. So kindly cooperate in that regard. Yes. The time has come now for me to propose vote of thanks. It gives me immense pleasure this evening to propose vote of thanks to our eminent guest joining us in this National E-Seminar on recent trends in plant sciences. Listening to the beautiful talks has really been an enlightening experience for all of us. On behalf of Sir Syed College, I thank our esteemed chief guest, the friend, philosopher and guide, Dr. Meenakshi Mahajan ma'am from Ferguson College, Pune. Madam, your presence here today means a lot for all of us. Thank for all you have done. It's also gratifying to see the guest of honor, the President, Raebarh Educational, Cultural and Welfare Society, Dr. Shamama Parveen. She has spared her valuable time to be with us today. Her support and inspiring guidance to our institution are of immense value. I pay my sincere thanks to the keynote speaker, Dr. Surendra Singh, Professor, Department of Biological Sciences, Rani Durgavati University, Jabalpur, for delivering the informative keynote address to cover the overall theme of the seminar. I hope all of you will join me to share the privilege of wishing him a long, happy, and prosperous life. We are grateful to the resource persons and the speakers, Professor Narayan Pandure from Department of Botany, Dr. Bamu, Professor K.K. Shao from the School of Studies in Biotechnology, Raipur, and Dr. Vasant P. Mali, Principal J. Vatumul Sadubule Girls College, Ulhasnagar, for they have given their valuable talks and suggestions to grace the occasion. I express my sincere gratitude to the Principal, Dr. Sheikh Kabir Ahmed, for his spirit of cooperation. We offer our wishes and greetings to the convener of the seminar, Dr. Milind Jadhav, and the co-conveners, Dr. Sangeeta Ahuja and Dr. Sangeeta Sutar, for the success of the seminar. We bestow our gratitude to the teaching and non-teaching staff of Sir Syed College for the generous support to make this National E-Seminar a big success. Last but not the least, we appreciate the sincerity and enthusiasm of the dynamic students and the researchers from various states of India. On this occasion, on this auspicious occasion rather, let us not forget to the contribution and sacrifices of the founder of Sir Syed College, late Professor Muhammad Tilawat Ali, in taking Sir Syed College to the appreciable heights. May his soul rest in peace. Thank you all for allowing me the time. May you all have the most enjoyable time. Thanks, one and all.
Thank you so much. Thank you so much.
We hereby declare the conclusion of the session. Thanks for joining us.
सर YouTube वर टाकलं YouTube वर
Thank you very much to all of you.
Thank you so much. Thank you.